Hello and welcome to this interview on Oncology Central. My name is Jade Parker. Today we are interviewing Professor Alejandro Madrigal. The interview will be on stem cell transplantation and his role at Anthony Nolan. Could you tell us about your career to date and current role at Anthony Nolan?
I am originally from Mexico. I studied medicine in Mexico and I did my training in internal medicine. After that, I went to Harvard University in Boston. I spent 3 years and a half there doing a kind of clinical postdoc. Then I came to England, to UCL, and I did my PhD here. And after 4 years, I went to Stanford University for another 4 years, and I joined the Nolan in 1993. And I became scientific director of Anthony Nolan in 1996, and I have been here since then.
Thank you very much. Could you tell us about Anthony Nolan's research strategy?
Anthony Nolan, as such, as you know, is a major registry. It was the first registry established in 1974 by Shirley Nolan, the mother of Anthony. And since then, it has played a key role in the community of unrelated bone marrow donation. Currently, there are over 30 million donors, and we proudly feel that this institution started the whole process of registry and become an unrelated bone marrow, the culture, let's call it like that. The Anthony Nolan Research Institute was established, as I say, in 1996 with the aim to improve the outcome of transplantation. So the whole strategy has been focusing ways to make the transplant more efficient, to reduce the complications post-transplant.
The work carried out at Anthony Nolan Research Institute focuses on improving the outcome of stem cell transplantation for patients with leukemia and other blood conditions. Can you describe some of the studies that the Institute is currently carrying out?
As I mentioned, in its short career, Anthony Nolan Research Institute has contributed significantly in the field of stem cell transplantation. We have published in the order of 1,300 papers that has been cited globally over 15,000 times. Our work has been very much focused in three areas. One is to improve the quality of the donor and the matching process between donor and patient. And because this, we have been fortunately to be collecting samples from donors and patients and do genetic and molecular analysis that has helped us to uncover the sorts of potential mechanisms by which you can provide a better matched donor. That has been a key role, and that has been very fundamental. In fact, this month we just published a paper that shows not just the fact that there are some factors like gene factors that we call it HLA, the major region that we try to match between donor and patient, but other factors like matching for cytomegalovirus in terms of the donor and the patient are positive or negative, and how that impacting the outcome. We also had focuses in cellular immunotherapy, trying to develop products or methods to reduce the main complications that happen post-transplantation, which are relapse, graft versus host disease, and infection. So we have been focusing in trying to develop products that help us to do that, or manipulate the immune system in a way that we can reduce this. We also have some component with the clinical translational research, which aims to bring many of the cord blood products. We have a cord blood bank, and we have our major research focus on obtaining cord products, cells, and sera and other factors that could help us also to improve the outcome of transplantation.
The Institute recently launched third-generation sequencing for HLA typing. Could you tell us about how this technology has improved donor-recipient matching?
So one of the major challenges that we had facing, and we in the community of transplantation, is to uncover the enormous, we call it polymorphism, diversity of the HLA genes. And the technologies that we had in the past has allowed us to see part of it, in particular when we do large-scale typing for unrelated bone marrow donation. With the new generation sequencing, we can actually uncover this difference. With the third generation sequencing, this is even more powerful. It's more easy because it allows to do full-length genomic amplification of the region that we are interested, and then screen and sequence those genes. So it gives us the real picture of how is the polymorphism in this individual and the match with the patient. We had now implemented in the routine, and we are in the process of studying the impact of this technology in terms of donor selection. We have about 1,200 donor and patient samples that we are in the process of analyzing and compare the impact of those differences that we find in the outcome. And we will tell you very soon. We believe that the better and the closer you match, the better is the outcome of the transplant. But we know also that when you match perfectly, there is a higher frequency of relapse. So we potentially are exploring the possibility to uncover what we call permissible mismatches, permissible difference that will help us to selectively provide more impact in terms of reducing the effect of what we call graft versus leukemia. So by doing that, we can eliminate the residual tumor cells.
In your opinion, what have been some of the most significant breakthroughs that have aided the advancement of stem cell transplantation?
I think we had seen a dramatic change in the field, in particular in the last years, not just with the stem cell transplantation, but with the uncover or the implementation of genomics and the manipulation of cells by using gene transfer that help us, for instance, to target specifically tumor targets for what we call CAR T cells. We also now have ways by using monoclonal antibodies to activate sleeping T cells, reagent cells that are capable to eliminate tumor cells, and by doing that, being capable to target specifically the tumor cells and reactivate the immune system to fight against them. In the stem cell transplantation, we also have obviously going from using bone marrow to mobilize stem cells, and then changing the protocols that has helped us to reduce the toxicity associated to the transplant. So that has been also a very critical issue that helps us now to transplant patients that in the past wouldn't be offered this procedure. Patients over 60 that before were not accepted, they didn't have a chance to be transplanted because the toxicity was so high that these patients will potentially die. Now we have this capacity to reduce the conditioning, and by doing that we can extend the benefit of this procedure. We also have seen the emergence of cord blood banks, and the use of cord blood stem cell transplantation has been very beneficial because it has helped us to target communities or individuals that in the past they didn't have donors in this pool of donors, because it seems that cord blood tolerates more mismatches. Cord blood transplantation allows you to perform it despite the fact that there might be some mismatches, and the outcome still can be as good or better than stem cell transplantation from unrelated bone marrow donors. And more recently, we had seen also modifications in the way we do haploidentical transplantation, what we call haplo-plus. By adding the post-transplant some chemo, you can reduce the alloreactive T cells, and by doing that, you can use highly mismatched individuals, mainly family members of the patient, and by doing that we practically could say that every patient now has the chance to be transplanted, has a donor, either bone marrow, unrelated, related, cord, or haploidentical. So that has been a major impact, that has made a major impact in the number of patients that can be transplanted.
On the flip side, what obstacles present the most significant problems with regard to improving patient outcomes?
As I mentioned now, practically every patient can have a donor, can be transplanted. But still, what we need to be sure is that the outcome is a good outcome. 50% still of the patients that get transplanted die, and they die, as I mentioned, from these three conditions. So we need to improve the mechanism to provide the optimal donor, whatever it is a perfect match or a perfect mismatch, we could use that terminology, and use cell therapy, immunotherapy, to remove the residual tumor cells to fight against infection and to reduce graft versus host disease. Tolerance is one of the major challenges. If we can extend the benefit of this procedure to other patients, reducing the toxicity, then we could be transplanting patients with autoimmunity. We already are doing that for some conditions, but we could extend it even with patients with lupus or other conditions. But we need to learn how to manipulate more the immune system in a way that it reduces toxicity associated to it. And we can actually fight more specifically either tumor cells or normal cells that we want to eliminate.
And how do you think these issues can be addressed in the future?
I think we are already working towards it. I think the collaboration and international community working together are targeting and developing methodologies that are, as I say, moving very fast. But still, we obviously with the new sequencers, with the next generation, with the new ways to understanding matching, because this is going to be a new challenge. We are going to uncover a large diversity of these genes, so we need to understand what we need to match and what we can mismatch. We need to also be able to produce specifically target cells and reduce the bottlenecks that at the moment limit the capacity that we have to give that to every patient. So I think we are moving towards that, but still we need to identify, for instance, more specific, what we call tumor-specific target motifs. And also we need to understand how we can manipulate the immune system, for instance, generating tolerance using Tregs or other cells that will help us to manipulate in a specific way the immune cells that help us to do that.
Do you have a particular direction that you would like the Institute's research to take, given the developments in the field?
I think we had recently reviewed our strategy and our research, and is focusing mainly in these areas that I had mentioned. We need to do more clinical translational component, and we have now developed with the community in UK a project which is called Impact, where we will focus in a way to translate more the research that we and other groups are doing to the benefit of the patient. I think in the next years the most important part is to bring all these innovations, cell therapy, in a more sort of broad way. So I think we are working towards that.